So our next speaker is Dr. Richard Novak. He is the co-founder and CEO of Unravel Biosciences, and Unravel partnered with Mito and over 20 families to analyze 4 different Leigh syndrome causing genes, and their results show some drug candidates that may benefit multiple Leigh syndrome patient groups, independent of the causative gene. So he's going to talk about unraveling these therapeutic candidates by partnering with patients, all of you watching. Thank you.
Great, thanks so much. Can you hear me okay?
Yep.
Perfect. Oh, it's a pleasure to be here. Thanks so much for the invitation to speak, and really grateful for the Mito support. So I'll be talking about Leigh syndrome, but really even beyond that, how we can, as we like to put it, defragment rare disease by finding shared therapeutic mechanisms. So thanks so much again for being here. And I really want to start with effectively the diagnosis, or sometimes lack thereof. And we're very mindful of the very unpredictable diagnostic odyssey that many patients face, especially in rare disease, where, of course, it starts with some sort of first symptoms and then a very convoluted sort of symptom management path later. You might get to genetic testing. Maybe there is a rare disease that's genetic in origin. Many times there's no diagnosis. In reality, it's about 60% of patients don't actually have a diagnosis. But you may have a diagnosis, right? So congratulations. And I think that's becoming, fortunately, more and more common. But yet, it might still take months or years to get there. So you think that now you have a diagnosis, you can actually have treatment. Well, sometimes that's great. And there is an approved treatment that actually works for you. But the reality is fairly grim. You might not have an approved treatment. It's sort of back to this symptom management kind of roller coaster. And that's unfortunately in about 95% of patients. And even if there is an approved treatment, sort of shown in formal clinical trials to be safe and effective, it may not work for you specifically. And so there's that personal aspect that may, cause the same problem. So what can we do? And so we started Unravel about 4 years ago with really a way to change this, and really do drug development driven by patients. The idea, let's incorporate this predictable medicine approach, really bringing engineering principles to drug discovery. And you may be familiar with some of these sort of logos. Here's, drug in the market, great. But here's a target. Usually a target starts at the beginning of a drug program, then goes into animals, and then goes into people. We basically said, well, let's start with the problem at hand. It's the patient's experience, not that of a mouse, not that of a theoretical target. And let's have that data really drive the drug development process and make this actually far more predictable. And the rationale for this goes back about 200 years. The whole idea is that it's individual patients, not diseases as an amorphous concept, that respond to treatment. And this goes back to William Osler about 200 years ago, saying the good physician treats the disease, the great physician treats the patient who has the disease. There's actually a view of our office that shows really how much we pay attention to that concept and value individual patients' journeys. And so what we're doing at Unravel is really changing how we go about drug discovery, and doing this by partnering with patients. And the concept behind our platform, and I'll talk about the technical aspects in a moment, is that we can use existing drugs as discovery tools. The ideal molecule may not exist today, and we may need to build a new molecule, but wouldn't it be great to know that it's going to work? And so that's where existing drugs, which could potentially be administered to patients through a clinical trial, could give us clinical validation. Yes, this target, this mechanism actually works in a real patient, and use that as a jumping off point for building even better molecules. Importantly, we can also defragment rare diseases. And I'm going to talk about that in a couple spots in a moment. But the whole idea is we've been trained to think that, okay, individual genes should have individual diseases, which should have individual treatments. And you're getting into this fragmentation problem. And biologically, that's probably what's happening. But from a commercial and drug development standpoint, that's the opposite of what we would like to do. And so it wouldn't be great if we can find treatments and shared mechanisms that work for a larger number of patients. Let's defragment as opposed to fragment the rare disease space. And then importantly, and this is something that is not something that Unravel does, but patients do need treatments today, not in 10 years. That new ideal therapy is going to take some time. But what can we do today as part of that discovery process? And that's really what we've done. And so we take a function-first approach. The whole idea is that we start with BioNav, this is the left piece here. And essentially we've modeled using various machine learning tools, health, and similarly a library of about 40,000 molecules, including a lot of repurposable drugs. Now, again, we are not a repurposing company, but these are discovery tools. And so we can start working with patients to essentially do virtual clinical trials. Then, of course, we have preclinical translation steps. Internally, we've developed what's called squishyware, which is essentially on-demand tadpole models, so little frogs. And we can, in a few weeks, make disease models of a whole range of disorders to very quickly ask, does it work in a sort of in vivo setting? So things like seizures, motor function, and even various cognitive functions can be tested. But then what we're always focused on is this right part, clinical studies. How do we know if it actually works? Well, until you test in real patients, not just models, but actual patients, you cannot answer that question definitively. Now, those preclinical models are key. And we also work with Jackson Labs and a number of other collaborators that are really fantastic at the preclinical translation side. But ultimately, we need to test drugs in actual patients. And again, this is where we accelerate the process. By using existing drugs, that can be done much faster and in a much less expensive way to get that critical clinical data before developing a new drug and having to go through that whole 10+ year process. And so just to kind of give a little bit of a background of what we did with Mito, the whole idea of this personalized predictable medicine comes into play, right? Individual patients respond to treatments. And so what we did, we used nasal swabs, just like the ones everyone's familiar with from COVID, to swab just the inside of your nose through the mail, so we can ship these all around the world and get RNA from patients and their matched controls wherever they are, so they don't have to travel, analyze that using our BioNav network analysis. And then the outputs are really, and what's going on with that disorder? Are there possible other disorders that sometimes happens? Predicting drug response to any number of therapeutics virtually, so there's no risk to the patient, and then working with clinicians to treatment options or some sort of general strategies with a foundation for clinical translation. And we found the last year really represent 8 states. And so it's a home collection kit. It's approved by the FDA for self-collection. And so it's minimally invasive. There's also no need for a cold chain. And it enables global sample collection very, very easily. And importantly, the nasal swabs, it's sort of an odd tissue. It's extremely complex. It's not skin. Sure, it's not the brain, but again, you don't really have people clamoring to give brain biopsies. So everyone's a little bit at a challenge with neurological disorders. And we found that nasal swabs actually provide a very, very diverse tissue source, including neuronal projections, by the way. So we do have some neuronal tissue. And we can detect really the vast majority of coding and non-coding transcripts. And so again, this kind of gives us a really rich source of data that we can then analyze. And importantly, we can work with wherever the patients are. And so one challenge with rare diseases and fragmentation that we see is geographic fragmentation. So if you don't live near a center of excellence, you may not be able to easily participate in clinical studies. And so you can participate through the mail. It makes it much easier and faster. And just to show how this process works, so this is actually not with patient data, but showing really the value of BioNav and what we can predict really without knowing anything about a disease. We can still narrow down on clinically meaningful therapeutics. So we did an analysis of tuberous sclerosis complex data, again from publications linked there in the bottom, and it was from 10 tuberous sclerosis complex patients and some control organoids, and these are brain organoids. And we looked at the RNA, ran it through BioNav, and we basically said, how do we fix this? How do we convert a patient organoid into a control organoid? And lo and behold, in the top few percent of the hits, about top 5%, were a drug class called mTOR inhibitors. And these are used absolutely as a first line of treatment for tuberous sclerosis complex. So it was a really nice validation. It has a very direct mechanistic rationale. And again, that took many years to uncover. We found this with maybe an hour or two of computation. And then on the clinical side, right, so do these predictions actually translate to patients? And so we work with a lot of really, really, unfortunately, severe cases. And in this case was a severe corneal de Lange syndrome patient. And this patient about a year and a half ago had status epilepticus, was unable to walk, and was really deteriorating over the last 18 months, spent over 100 days in the hospital. And they decided to share our results with their clinician, and the clinician actually identified a couple molecules that they were willing to prescribe. And really, these are more in the supplement range, so nutraceuticals and a couple, fairly modest prescription drugs. And within 7 weeks, the patient was able to walk on her own for the first time in a year and a half, and continues to improve. And so what we've done is over the last few years built a new business model. So we have two parts of our company. Both are for profit, but one is more of a not-for-profit service platform, which is called RareShift. Essentially, foundations really needed a way to generate data, identify potential repurposing paths, and that's what we tailor this for. But then as we generate that data, we're always looking at, well, what do the new drugs look like? What new mechanisms and targets should we be looking at? And that's the biotech arm of our company. And so to date, we have over 70 different programs, all at various stages of development. And so here's just a snapshot of some of them. We are actually about to start 2 clinical trials. We're in the final stages of regulatory approval for just kind of final, going back and addressing the last remaining points. And that's for Rett syndrome and Pitt-Hopkins, same drug, but used for 2 different disorders. Another one in the Philippines that Mass General Hospital is working on, and several other groups that were actually developing observational studies, and a number of off-label testing that was done by the patients with their clinicians. And we've been working with Combined Brain to really dramatically amplify the reach of our approach, working there with over 120 different disorders, and then with starting to work with children's and cancer hospitals to provide that additional research data, that personalized data to drive their approaches. And so now what have we done with Cure Mito? So the whole idea was, let's find potential treatments for Leigh syndrome, obviously preferentially repurposed ones. So again, grateful for the funding and the support and the collaboration with actually a number of other researchers that were likewise funded. And so the purpose was to generate clinical data, analyze the population, what does the population look like, and then identify potential treatments, right? So relatively straightforward. So we have, we started with 18 Leigh syndrome patients, and then we added 4 other mitochondrial and sort of ATPase patients that really have some overlapping symptoms. And there is this concept of kind of ATP, disruption. And we want to see what would happen. And then we have basically the matching same-sex family members. And so really grateful to the direct patient and family partnership. This is what drives the science. And so, long story short, just as an aside, we as a policy do not share drug names in public presentations, but certainly the foundation has all the data. But this is a correlation plot. So these little squares in the middle is basically each of these lines or columns is a patient, and basically an individual drug prediction for that patient. And we want to see how is that list of drugs from most therapeutic to essentially anti-therapeutic. How does that correlate with every other patient? And so you have sort of a 18 by 18 patient matrix here. And the more red it is, the more positive it is, the more similar the drug lists are, the more blue it is, the more opposite it is. So what's good for one patient is actually bad for the other, and so on. And so we saw about 2, sort of a major group and a minor group. And importantly, this covers 4 genes. So ECHS1, ATP6, ND1, and SURF1. There's no clear pattern in terms of the sex, the age, and other aspects of this disorder. And yet we got some very, very strong clustering here. When we add in several of the other patients, and now it's 8 different disorders, including LONP1, ATP8, ATP1A3, ATP5, so some more mitochondrial, some more just ATPase. We were more curious to see what would happen. We still get 2 groups, and again, about 2. It's sort of 2.5, but not that dramatically different. And again, there's no obvious trend here. But so we want to say, well, what separates these out? And looking just at the causative genes, we want to see, okay, is there any sort of clear difference just in terms of the genes that do cause these disorders individually? And overall, there isn't, except for ECHS1. It looks like actually ECHS1 expression, even though not most patients don't have ECHS1 mutations at all, just the level of it seems to correlate with those 2 different subgroups. So there seems to be a trend that was worth exploring. Essentially, it's a marker for separating out these groups, at least one of many. And then we wanted to see, to make this predictable and actually do have a drug development path, you need to know what that path looks like. And it always takes some sort of preclinical model. And so what we did is we collected several public data sets all around SURF1 models. So there were iPSC-derived just 2D lines. There were cerebral organoids, again, SURF1 mutants, brain, liver, and fat from a mouse, and then a sort of younger iPSC model that was actually the same as that one. And what's really exciting is that you still have these kind of two and a half groups, but these different models align with different subgroups. And so it's not just that all SURF1 patients are mimicked by the same SURF1 model. For example, even the age of the iPSCs, it's either group 1 or group 2. It's not, they're not all together. And what's interesting, the mouse model actually clusters really nicely with this sort of subgroup here. And so the whole idea is that, even for the preclinical drug development, we don't want to have to test a therapy in 8 different models if we're going to go into 8 different patient groups or disorders. And so again, but how do we separate them out? And so the whole idea was, let's look at these various markers. And so this is a gene list that we looked at, basically all these Leigh syndrome associated genes from this 2014 publication. Again, we can look at any other gene, of course, but we wanted to limit it. And we wanted to see which genes are significantly separating out these subgroups. And so we were calling sort of group 1, group 2, and 2.5. So 2 and 2.5 are depending on how you look at the numbers, either one group or 2. For any one drug, it's really 2 groups. And we wanted to see how would we be able to identify patients if we go back. If I have a patient, do I have to go through this whole AI drug generation process, which is much more complex and more expensive, or is there sort of a handful of markers we can use to identify patients? And again, it's going at the concept of, are there similarities across different disorder groups? And so that's what we did. These are actually all significant genes. All of these are mitochondrial genes. And so there are these patterns that are starting to emerge. And if we look at any genes altogether, they really span cell adhesion, the extracellular matrix, ciliary function, and morphogenesis. And again, there's a whole big section on ion transport that's more diffuse. So again, there are these markers that we're finding. And essentially where we're at right now, we generated this new RNA-seq data that's available for analysis. We've identified these interesting subgroups and potential markers to stratify them. And there's a number of clinically translatable drugs, including the azoles that Dr. Prigioni talked about. So really interesting path here in terms of what's next, working with clinicians to look at what do these preclinical models look like? How do we stratify the models to the patients? And then ultimately, what does that clinical trial look like? And I just want to leave you with this parting thought around defragmenting rare disease through this predictable path. The whole idea is that we're looking not only at Leigh syndrome, not only at mitochondrial disorders, but really at our whole database of many hundreds of patients. And this is a project that we've been working on internally. And so it's 48 patients shown here, 10 different genes. So this actually includes quite a number from CSNK2A1 and SYNGAP, which are not mitochondrial disorders. And they still cluster into these 2 to 3 groups. So leaving us with this concept that, by developing 3 drugs, we may be able to target many thousands of patients as opposed to a few hundred at once. So thank you again to Cure Mito. And I'll just stop here with this concluding slide. Thanks to the Unravel team for making all this work happen and for the wonderful partnerships with patients and their families. Thank you.
Great. Thank you, Dr. Novak. I know the collaboration between scientists and families and personalized medicine are concepts that resonate so much with this community. So we appreciate Unravel's efforts alongside Cure Mito in this realm.